ಇದೊಳಗ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ಸ್ ಆಲೋಚನೆ ಒಲೆಯನ್ನು विटिकॉसल
स्टार्ट हो गया
अलीशा अलीशा को विटिकॉसल
लाइव चल रहा है
मिलिट है ना
So I'm going to be presenting an article on harnessing the anti-cancer natural product of Nimbolite for targeted protein degradation. So this article was published by the author is Jessica N. Spradlin. So the study's aim is to investigate the mechanism of action for natural product Nimbolite, which is derived from the neem tree, across the world. Nimbolite is shown to inhibit tumorigenesis and metastasis without causing any toxicity or unwanted side effects. In previous studies, it was seen that Nimbolite impairs cancer pathogenicity by modulating signal pathways and transcription factors that are linked to survival, growth, invasion, inflammation, and angiogenesis. It has been difficult to identify the direct protein target of natural products, which is why this article will be trying to use activity-based profiling, protein profiling, ABPP, chemoproteomic approaches to map the protein-wide targets of Nimbolite in breast cancer cells proteins. So the method of action of using the ABP approach, it was found that one of the primary targets of Nimbolite breast cancer is cysteine-8 of the E3 ubiquitin ligase RNF114. Nimbolite covalently modifies RNF114, which leads to impaired ubiquitination and degradation of its substrate, which is the tumor suppressor CDK1A. This inhibition happens through impaired substrate recognition, which may indicate that Nimbolite can be used as an E3 ligase recruitment model for protein target degradation. ABPP is activity-based protein profiling. So this is the chemical structure of Nimbolide. This cyclic ketone pair is cysteine reactive, so that aids in targeting E3 ubiquitin ligase RNF114. So in this experiment, there's a study done with serum-containing media and serum-free media, where they were both treated with Nimbolide, and the study was focused on triple-negative breast cancer. The reason why they chose this is because TNBC are aggressive forms of breast cancer. So the results of this experiment showed that Nimbolide impairs cell proliferation in both situations. So here you can see as the concentration increases from 1 to 10 to 100, this percentage of proliferation decreases in both case scenarios. So another study was done using flow cytometry. 231 MFP cells were treated with Nimbolide for 24 hours and then 48 hours. FITC annexin 5 and propylium iodide are fluorescents that are used. This one detects dead cells. So the results showed that there's a significant increase in apoptotic cells after the Nimbolide treatment. Sorry. So another technique was used to figure out exactly what the target protein was. Isotope ABP, which is a chemoproteomic platform called isotopic tandem orthogonal proteolysis enabled ABPB, which is used to determine the specific target of Nimbolide. This technique uses reactivity-based chemical probes that map reactivity, functional, and ligandable hotspots for in complex proteins. So this is sort of the overview of the approach. They use control and the Nimbolide treatment. So they start off with labeling the cells with cysteine reactive alkyne functional iodosis probe, and then they enrich the labeled proteins with heavy and light isotopes so that they can further analyze it in the LCMS-MS. So the results of this experiment said that there was one primary target, which had an isotopic ratio of greater than 4, and it was significantly engaged by nimbolide. This was the E3 ubiquitin ligase RNF114. So it concludes that RNF114 contributes to the anti-proliferative effect of nimbolide. This conclusion was further demonstrated when they used RNF114 knockdown to figure out that it actually showed the opposite effects. So it showed the resistance to anti-proliferative effect of nimbolide. Knockdown, it's like, from what I know, I think it's when they take out the gene that
What is
the knockdown for electrolysis.
whether this RNF114?
So RNF114 is the enzyme that helps nimbolide have its effects on anti-cancer. Thank you.